Good morning everyone. Today we will discuss about DNA replication. In the previous lesson I have taught you about semi-conservative mode of DNA replication and experimental proof of the DNA replication, which was given by Michelson and Stahl. As you know, the process of DNA replication takes place during the S phase of the cell cycle, or you can say during the process of cell division. The DNA duplication or DNA replication takes place in synthetic phase. And DNA replication is very important. When one cell divides, the DNA amount is going to be duplicated, and thus the DNA is transferred also from one cell to the another cell. So DNA replication, what is DNA replication? DNA replication is the process of the synthesis of DNA from the DNA template. It means the DNA gives rise to the new strand of the DNA. And as you know, when the DNA is replicated, suppose this is the DNA strand, or you can say duplex DNA, it has two strands. At the time of DNA replication, both the strands are separated to each other, and these are the old strand. This is also old strand, which act as the template, and as per according to the old strand, here the new strands are synthesized in 5 to 3 direction. Because the new strand, so out of 2 strands which is synthesized newly, one strand is parental and another strand is newly synthesized strand. And this kind of the DNA replication or DNA duplication is said to be what? Semi-conservative mode of DNA replication. So once again, what is semi-conservative mode of DNA replication? In semi-conservative mode of DNA replication, in a newly synthesized DNA duplex having one old strand and one strand is newly synthesized. One is new and one is parent, or you can say is old. And this is called semi-conservative mode of DNA replication, which I have already explained. Now, in the case of DNA replication, or you can say during the process of DNA replication, so many enzymes are required. The proteins are also required, some of the primers. So what are the materials which are required during the process of DNA replication? First of all, enzymes, and these enzymes are like DNA polymerase, you can say also DNA dependent DNA polymerase enzyme. Second, helicase, then topoisomerase enzyme. The next one is the RNA primase. Some proteins, DNA, one more enzyme, DNA ligase. I will explain the function of each enzyme during the process of DNA replication. But first of all, you see that how many enzymes are required for the DNA replication. And I told you protein is also required during the process of proteins, and these proteins are SSB protein. SSB protein means single strand binding protein. After the protein, then the substrate is required, and these are deoxyribose nucleoside triphosphate. Deoxyribose nucleoside triphosphate, these all are the substrates. Then you will see the RNA primer is also required, RNA primer, and RNA primase I have already written here. So these all are the materials which are required during the process of DNA replication. I will explain the function. The DNA replication starts at a particular point. At a particular time, not whole of the strand is going to be separated to each other. For the process of replication, it takes place on the DNA strand at the specific position, and that position is called origin, or you can say origin of replication. It is very, very important. The origin of replication is the site on the DNA duplex on the DNA molecules from where the DNA replication is started. That is called origin, or simply you can say origin of replication. During the process of DNA replication, first of all, you will observe that the duplex DNA, you can see here, this is the diagram. Or you can understand here, suppose this is the double-stranded DNA. This double-stranded DNA, first of all, both the strands, they are separated to each other. It means the hydrogen bonds between the nitrogen bases, between the nitrogenous bases, they are degraded, and due to the breakdown of the hydrogen bonds, both the strands of a DNA duplex, or you can say both the strands of DNA molecule is separated to each other, and it makes a replication fork. It's very, very important. So a particular enzyme is required. The name of this enzyme is called helicase. The helicase enzyme, the first this enzyme helicase enzyme unwinds the duplex DNA to make the DNA template, to make the replication fork. What do you mean by replication fork? When the duplex DNA, or you can say the double-stranded DNA, both they are separated to each other, it forms a Y-shaped structure, and that is called replication fork. And the replication fork is the position, is the place where the process of DNA replication is started. And this replication fork is produced by which enzyme? Helicase enzyme. So this structure is called replication. This is the form. And each strand, this strand act as the template. This you can say DNA template. This is also the template. And the polarity of this strand, this holds 3, then it will be the 5. It will be 5 end, then it will be the 3 end. At the time of the formation of the replication fork, when the duplex DNA is separated to each other to form the replication fork, this Y-shaped structure, in the presence of helicase enzyme, upper side or the duplex DNA is going to be supercoiled. Means when the DNA strand, the double strand DNA is going to be separated to each other in the presence of helicase enzyme, the upper side, the duplex DNA is going to be twisted again, and this is called supercoiling. If you will take the two ropes and both the ropes, they are wind to each other. If you are going to unwind, you can see the twisting again, and this is called supercoiling. And this supercoiling is relaxed by a particular enzyme, and that is called topoisomerase. So I can write here one more enzyme is called topoisomerase. This enzyme, the topoisomerase enzyme, is required to relax supercoiling of the duplex DNA. So helicase enzyme is responsible for unwinding of the duplex DNA, and during the process of unwinding, the supercoiling takes place, and that supercoiling is relaxed by the enzyme, and that is topoisomerase. Now here some proteins are required during the process of DNA replication. You can see when the unwinding takes place, when both the strands they are separated to each other, a single strand is being holded by some of the proteins, and these proteins are called SSB proteins. SSB proteins, single strand binding proteins. The SSB proteins binds the single strand of this duplex DNA, and therefore the duplex DNA or double-stranded DNA is not going to again join with the hydrogen bonds. If they are not going to be holded by the SSB protein, they may be coiled again, and there may be winding of the duplex DNA to form double helical structure again. So when the replication fork is formed, or you can say when these two strands they are separated to each other in the presence of helicase enzyme, a single strand, or you can say these two strands which are single, these strands are holded by a particular protein, and that is called SSB proteins, single strand binding proteins, which binds the single strand and prevents the binding of duplex DNA. Now you will observe from these two strands, now each strand, when the duplex DNA is unwinded or both they are separated to each other, the single strand now act as the template. What is template strand? The template strand is the parent strand. This is also template, this is also template, and as per the template strand, you will observe the new strand will be synthesized. You will observe like this. And here, because here the base complementary rules are very, very important. As you know, the substrates are required when the process of DNA replication takes place. So many nucleotides, deoxyribonucleotides, are required, and all these nucleotides, they are going to be arranged, they are going to be polymerized in the presence of DNA polymerase enzyme as per base complementary rules. So as per the DNA template, suppose in the DNA template here a particular nucleotide is to be there, that is adenine. As you know, adenine, it combines with the thymine, so here the nucleotide, it will come thymine. If here there will be the cytosine, then it will come guanine. If on the template strand there will be the guanine, the nucleotide having the guanine, then it will become cytosine. If the thymine, then adenine. So as you know, as per the base complementary rules, according to the DNA template, the new strand which is complementary to the old strand, it is going to be synthesized. So which substrate is required? That is called deoxyribonucleotides, or you can say deoxyribose nucleoside triphosphate. And this substrate, deoxyribonucleoside triphosphate, this substrate performs dual function. The first, the triphosphate, deoxyribose triphosphate, act as the substrate, and it also provides energy for the process of polymerization. So it has dual function. Deoxyribose triphosphate has dual function. The first one, it acts as the substrate in the process of DNA replication, and also it provides energy for the process of polymerization. The process of polymerization takes place by an enzyme that is called DNA dependent DNA polymerase enzyme. The DNA polymerase enzyme, simply you can say, this enzyme is very, very important for the process of polymerization. And the polymerization rate is very high, nearly 2000 base pairs of the base pairs size is going to be polymerized only in a very short duration of the time, within a minute. So DNA polymerase enzyme has high polymerization rate. Different types of the DNA polymerase enzymes are present in the case of prokaryotic cell, but all these types are not included in the NCERT book. You just have to focus that what is DNA polymerase enzyme. The DNA polymerase enzyme is a DNA dependent DNA polymerase enzyme, and this enzyme is required for the process of DNA polymerization, or you can say for the addition of the nucleotides according to the template. And DNA polymerase enzyme also helps in proofreading. Suppose if, as per DNA template, here some of the wrong nucleotides are going to be added during the process of DNA replication, it also prefers editing, or there is the proofreading also. It goes on this direction, again it comes back and check if any wrong nucleotide is being added. It can remove, it can replace by the correct nucleotide. So it also helps in the proofreading, in editing of the nucleotide, whether it is wrong or correct nucleotide. So DNA polymerase has how many roles? The two roles are there. First, it helps in the process of DNA polymerization, that is, addition of the nucleotides as per base complementary rules, and second, it also helps in the process of proofreading. Now, it's very, very important when the polymerization takes place, you can see here the DNA polymerization or the new strand is synthesized on the template only in which direction? In 5 to 3 direction. If there is a question there, what is the direction of the synthesis of the DNA? That is 5 to 3 direction. And what is the polarity of the template strand? The template strand polarity is 3 to 5. So, as per 3 to 5, in this direction, this will be the 5 to 3 direction. And suppose here, this is 3 to 5, then again in this direction, the new strand will be synthesized. So this is the direction, the new strand, this is synthesized on a single strand on this DNA template. On this side, you can see here, the new strand is going to be synthesized in this direction 5 to 3. Again, this is also 5 to 3. It will not start from here, otherwise the polarity will be the same. DNA polymerase has one drawback. The DNA polymerase enzyme do not synthesize, do not initiate the polymerization. It can't start, it can't initiate the process of replication de novo. It needs a primer, and without the primer, the DNA replication is not started. And this primer is called RNA primer. So first of all, it is not given in the book. A primer is required here, and this is called RNA primer. The RNA primer also has, this one is the 5 polarity and this will be the 3. And then primer, it gives, or you can say, it helps to start the further polymerization in the presence of DNA polymerase enzyme. And this primer, RNA primer, actually RNA primer is the short chain of the ribonucleotide, and this short chain of the ribonucleotide, that is RNA primer, is synthesized in the presence of an enzyme that is called RNA primerase. It provides the 3 end, and again here the deoxyribonucleotide, their 5 end joins with the 3 end, and it forms the phosphodiester bond. Once the chain is started, again the primer is being removed. It is going to be replaced by the deoxyribonucleotide. So without primer, primer just starts the process of DNA replication, and later on the primer is replaced by the deoxyribonucleotide substrate. You can see here the DNA polymerase synthesized the strand on one template. On my left side you can see here, this is called the continuous strand, because here both the strands, they are going to be as the duplex, this is the duplex DNA, double stranded DNA, both the strands, they are going to be replicated simultaneously. So on one strand, the DNA replication is continuous. This is called continuous strand. Continuous strand, and this continuous strand is also known as leading strand. While on the another DNA template, the DNA the new strand which is synthesized is not continuous. You can see some fragments are to be there, and these are called discontinuous strand, and this is also known as lagging strand, or this strand was actually reported by a scientist Okazaki. That's why it is also known as Okazaki fragments. So, when the two strands are synthesized on the DNA template, one strand, you can see here, this is the continuous strand. The continuous strand is also known as leading strand. On the another hand, on this template, the DNA strand which is newly synthesized has short stretches of the segments, and these are called Okazaki fragments, or you can say discontinuous strand, or this is also known as lagging strand. But further, the Okazaki fragments are also joined or going to be attached by which enzyme? DNA ligase enzyme. So DNA ligase enzyme helps to join these Okazaki fragments, or you can say the lagging strand, and finally it also becomes continuous. So that's all about the process of DNA replication. When this strand, it opens, and it will be the old strand, and this will be the new strand. Again, this will be the old strand, and this will be the new strand. Both they are, again it forms the duplex DNA due to the hydrogen bonds. So one DNA molecule gives rise to two new DNA molecules, and this is called DNA replication. So in DNA replication, as you have observed, one strand is old, the another one is the new. This is old and this is new, and thus this is this all about the semi-conservative mode of DNA replication. So once again, you can see about the DNA replication, that DNA replication is the process of the synthesis of the new DNA molecule from the old one. The DNA replication is semi-conservative type. In semi-conservative DNA replication, the newly synthesized strand has one old strand, and while another one is the newly synthesized. During the process of DNA replication, so many enzymes are required. As I told you, for example, helicase enzyme helps to unwind the duplex DNA. So the replication fork, this is the diagram of actually replication fork, is formed. The topoisomerase enzyme helps to relax supercoiling. The DNA polymerase enzyme is the enzyme which is required for the process of polymerization. During the, what is polymerization? In the process of polymerization, as I told you, deoxyribonucleoside triphosphates, means deoxyribonucleotides, simply you can say, they are added to each other by phosphodiester bond as per base complementary rules. According to the template, the nucleotides are going to be added, and this addition of so many nucleotides to form a long chain, this is called polymerization, and polymerization takes place in the presence of enzyme, and that is called DNA polymerase enzyme. DNA polymerase enzyme, not only it polymerizes the new strand, it not only helps for the process of polymerization. Even if any wrong nucleotide is being added into the new strand, it also helps in the proofreading. It can replace the wrong nucleotide by the correct nucleotide in the strand. So it has two functions. The first, it helps in the process of polymerization, and it also helps to replace the wrong nucleotide by the correct one. Then when the DNA replication starts, the DNA polymerase enzyme do not start the new strand. It can't initiate the process of DNA replication. It always needs a base from where it can add so many nucleotides. That's why RNA primer is required, and that RNA primer initiate the process of DNA replication. And once the process of DNA replication is started in the presence of DNA polymerase enzyme, once some of the nucleotides are being added, the RNA primer is also being replaced by deoxyribonucleotides. And here the deoxyribonucleoside triphosphate has dual function. The first, it acts as the substrate for the polymerization, and second, it also gives the energy which is required for the process of polymerization. The polymerization, or you can say the new strands, are synthesized on the template one. On one template, the new strand is continuous, and this is called continuous strand or leading strand, while on another template, the direction, it is 5 to 3. This is 5 to 3, and on the another strand, it is discontinuous. So it is called discontinuous strand or lagging strand, and this lagging strand are also known as Okazaki fragment. The Okazaki fragments are lagging strand, or you can say the discontinuous strand, or further joined with the enzyme, and that is called DNA ligase enzyme. So DNA ligase enzyme helps to join the Okazaki fragments. The SSB protein, as I told you earlier, the SSB protein helps to hold the single strand, and thus it prevents the binding of the duplex DNA. So this is all about the DNA replication. All these enzymes, the protein, the substrate, and these components are very, very important, and you have to learn their function. So that's all for today.